Antonio de Maio. Doctor Antonio de Maio se graduó de la Universidad Central de Venezuela en Caracas. Posteriormente recibió su doctorado del Instituto de Ciencias Weizmann en Rehovot, Israel. Fue profesor asociado y director de investigación de la División de Cirugía Pediátrica de la Universidad Johns Hopkins. Dentro de las investigaciones de su laboratorio se encuentra el estudio de las bases moleculares y genéticas de la respuesta a la lesión. Actualmente es profesor y miembro del programa de posgrado en Ciencias Biomédicas en la Universidad de California en San Diego. Recibamos con un fuerte aplauso al doctor Antonio de Maio.
Para mí un gran placer que me hayan invitado a este meeting. Yo soy originalmente de Venezuela, pero México se ha convertido en mi tercera patria y eso me hace un gran honor estar aquí con ustedes. Me voy a disculpar porque voy a dar la charla en inglés, pero después de la charla podemos hablar todo el español que quieran. Tengo que disculparme porque mi español se ha contaminado un poco con la lengua de Tijuana.
Es todo, Manuel.
So I know that it's been a long morning and some of you are tired. So please pay attention because I'm going to repeat this only once. So I would like to start the talk with this phrase of Paracelsus that say that the body possesses the high art of wrestling and also restoring, meaning that the diseases that we produce are also forms to fight them by the organism. So cells and organisms are always constantly exposed to environmental fluctuations, causing changes in gene expression directly to preserve homeostasis. But in some cases, environmental changes may compromise homeostasis and cellular integrity, resulting in stress conditions. And since this is a clinical meeting, pathologies are also stress conditions. Now, cells and organisms are like political parties. Their mere function is to survive. And to survive, cells need to maintain, to restore homeostasis, which is accomplished at least in part by the efficient mechanisms for intracellular communication. So how cells communicate? There's different mechanisms. One, what is very, very well known is soluble molecules like hormones that are released by one cells, interact with another cells interacting with a receptor. There is also the exchange to low molecular way metabolized between adjacent cells by gap junctions. There are cell-cell contacts, so cellular synapses, or also transfer of membrane pieces between one cell and the other one. But there is a form of communication that is emerging with great importance, and is extracellular vesicles. So what are extracellular vesicles? They are membrane-encapsulated particles derived from the plasma membrane that carry components from the cells, including proteins and glycoproteins, nucleic acids, lipids, carbohydrates, and other metabolites. And extracellular vesicles can interact with target cells and modulate their function. So there is no one type of extracellular vesicles. I know that you know well what is exosomes. Exosomes is part of the extracellular vesicles. There are many different kinds of them. One of them are called ectosomes. And ectosomes are derived directly from the plasma membrane, maintaining the composition of the membrane. And these ectosomes come in different sizes. Some of them are small, 100 nanometers. Some of them are larger, so I call microparticles. There is also exosomes. So exosomes are produced by more complex mechanisms in which the plasma membrane is internalized and is directed to a vesicle, forming multiple vesicular bodies that later are released in the extracellular environment. And the importance of this process is that these vesicles, when they are released, contain the same topology that the surface of the cells, in the same way that the ectosomes. And it's also apoptotic bodies that are produced with cells are dying, and it's just a random release of vesicles from the cells of different sizes and different compositions. So we have defined the stress observation system and a way that extracellular vesicles act as sentinel molecules that prime other cells during stress conditions to activate a rapid response to avoid the propagation of the stress of insulin. And the idea is that during normal conditions, cells are producing extracellular vesicles that contain certain composition that demonstrate the physiology of the cells. And if there is no disease, there is no stress, these vesicles trigger the immune system and tell them everything is okay, nothing is happening. But if there is a stress or a pathological condition, the composition of these vesicles change. And now they can tell the immune system that something is happening and they take an action. Now, the composition of extracellular vesicles is likely to change during pathological conditions, and therefore they can be used as a marker of disease. And in fact, there is extracellular vesicles being detected in many pathologies, particularly in cancer. There is a very active field of research of extracellular vesicles, but there is many other vesicles that you can see in this table that maybe is going to be difficult to read. Now, extracellular vesicles could also be used as a diagnostics. And we all know that the best diagnostic will be the autopsy, but you are not going to go to that way. So, extracellular vesicles can be isolated for a variety of body fluids, like blood, saliva, and urine. Now, recent work in our laboratory have found that urine is an ideal body fluid for the isolation of extracellular vesicles, especially because it can obtain in great quantities and is painless. And we have developed a very nice protocol to isolate extracellular vesicles from human urine samples. And I'm not going to tell you the details of the procedure, but I just want to give you some idea of what is a remarkably interesting finding with these vesicles. So in our isolation, we do differential centrifugation. And what we did is, if we want to use these vesicles as a marker of disease, the first thing is to establish what is the baseline. So healthy people, how much variation you have in extracellular vesicles from urine in healthy people. So what we did is that we isolate vesicles from the same person twice a day for 5 days. And to calculate how many vesicles was in the urine. And what we found that was remarkably interesting is that the number of vesicles in the urine of this person was the same independently of the time of the sample or independently of the day. As you can see in this graph, there is about 1 million, 1 per 10 to the 10 vesicles per ml of urine. So 10 ml of urine can give you a lot of material to work with. Well, that was one person. So the other thing that we checked was what happened if you have different people. And this is 5 different healthy individuals. And we isolate the vesicles, and you can see that at least the number of vesicles is very consistent between everybody. And now we have some molecular markers that we can use to use base conditions for these healthy people. And now the challenge, and I invite all of you to participate in this, is to get urine samples from patients and try to analyze whether these change the number of vesicles in the sample or the composition of the vesicles, and try to use this as a diagnosis. Well, our real interest in the lab is how extracellular vesicles will activate cells. And as I showed you before, we have the stress observation system. So the idea is that these vesicles can live in place far away from the cell that is producing these vesicles, target cells at a distance and modify their function. Now, there is a problem with extracellular vesicles. The first is that they are very heterogeneous, even when they are isolated for a single cell type. So extracellular vesicles, when you take a pool of extracellular vesicles, they are not identical. And the composition of each one of them is very different. In addition, because they are so small, they only contain a very limited number of components. We have calculated how many proteins can you pack in 100 nanometers vesicles and found that you can get between 100 to 200 copies of a protein in a vesicle. That's a really very small number, and that's we're pushing the limits. We calculate how many microRNAs are very popular in extracellular vesicles, and you can find that there is less than one microRNA copy per vesicle. So there is plenty of vesicles with no microRNAs, a few of them with some. So when you think about how these vesicles can activate cells, you need to think that they are mainly for a cell to be activated, need to capture a large number of vesicles with different compositions, and they are, in a synergistic way, produce an action on the cells. And in fact, this is a slide that we show here, biotinylated vesicles. We took normal vesicles from the cell line, and we biotinylated them and incubated with macrophages. And in the first panel, you will see what is the cell surface, the binding of these vesicles to the cells. But when you permeabilize the cells, and you come with spectroscopy to see how many cells, you can see that the amount of vesicles that are captured from these macrophages is astronomical. And you need to think that each one of these vesicles have a different composition, and it's all of them producing an effect. Obviously, that tells us that working on this system is quite complex. But they produce a very strong reaction, and this is the same work, and we found that stress and non-stress vesicles can induce TNF-alpha and macrophages in a quantity that is just remarkably high. So the question was, so how can we discern how these vesicles are activating cells? Now, we thought that, well, there is one thing that all these vesicles contain, is a membrane. And the membrane is composed of phospholipids, mainly phospholipids and cholesterol. So we decided to investigate how phospholipids within membranes can activate cells. And a very simple method to analyze this is to try to mimic vesicles by using liposomes. So liposomes are made of phospholipids. So we make phospholipids with a specific composition of the side of the extracellular vesicle. So we do this, we prepare liposomes of two lipids that are the major components of the plasma membrane, phosphatidylserine and phosphatidylcholine, PC. And I want you to point out in this slide that we're using these lipids. The only difference is the head. The fatty acid of both lipids is the same. And we investigate their effect of peritoneal macrophages in vivo conditions. So this is very important. I want to make a point here. The reason that we did it in vivo is because we know the macrophages are very sensitive to the environment. And when you take macrophages from one place and put it in another place, like you take peritoneal macrophages and you put them in culture, they become different cells. The response in culture conditions is not the same as the response that they have in vivo. So we decide to inject these liposomes into the peritoneal cavity of mice. We use 10 to the 12 liposomes. We calculate about, we're given 5 to 10 to the 5 liposomes per macrophage, per peritoneal macrophages. and we use an injection of PBS as a baseline. And later on, we harvest the cell 1 hour because we want to know what is the immediate response. We don't want the liposomes to have a systemic response, and we want to see what happens immediately after these macrophages take the liposomes. So first I'm going to show you that indeed the macrophages take the liposomes. So we take these liposomes and we encapsulate them with AF647 dextran. So this is the lupin, highly fluorescent. And we inject it into the mice, and 1 hour after we harvest the peritoneal macrophages, and we analyze them by flow cytometry. And in the top panel, you can see that the majority of these liposomes are captured by macrophages and a very small amount by B2 cells. And in the bottom panel, you can see a cell line that also took these macrophages, so you can see that they are uptake. We believe that it's by a process of granulocytosis, and they accumulate vesicles inside the cells. So next, what we did is that we did a transcriptome analysis by doing RNA-seq. Our analysis with these peritoneal macrophages that were PC or PS stimulated or baseline with PBS, we found something really very interesting. About 4,000 genes were modified by just these two phospholipids. One of them were, 4,000 genes were affected just by by PS, and 1,500, 1,600 genes were modulated by PC. This is a remarkably high number of genes. In total, it's almost 5,000 genes that were independently modulated by both lipids, and 1,400 genes were commonly affected by both lipids. So to us, that was a real surprise, because now we're dealing with a single lipid in a liposome that is capable to modify a large number of genes. So we did analysis how many genes are specific for each one of them, and what we found is that indeed there is 1,400 genes that are specifically altered by PS in comparison with cells treated with PC liposomes or PBS. So what is this 1,500 genes telling us? So we did a single pathway analysis, and we found that there were 6 signaling pathways that were activated in this process and were downregulated. And the main pathway was the cytokine. Okay. So the most significant pathway was the cytokine receptor pathway. That is basically a pathway for inflammation. And we found that in this pathway, there were 27 genes that were upregulated specifically by PS and not by the PC or the PBS. Here is how these genes, all the 27 genes, I don't pretend that you read this, but just to show you a flavor of the regulation of the genes. Some of them are down-regulated with PBS or no regulated at all with PC. Red means high, blue means low, white means nothing. And here is the pathway. And I don't pretend that you see this in detail, but here we mark in red all the 27 different genes that were upregulated. And many of them have been validated by quantitative RNA, mRNA, by quantitative RT-PCR. And as you can see in the blue columns, that all these genes are specifically regulated by PS and no PBS. And to trace our system, we picked IL-10, that is another inflammatory gene, that was not in the pathway. And in the last slide, you can see here, between the orange and the red and the blue, that IL-10 that is not in the pathway is equally activated by PC and PS, demonstrating the robustness of our analysis. So among these 27 genes that were specifically regulated by PS liposomes, we identify several chemokines. And we know that chemokines are involved in the recruitment of neutrophils. Therefore, we predict that if we inject PS liposome in the mouse, it should be a recruitment of neutrophils in the peritoneum. And indeed, that's what happened. When you inject PS liposomes and now you harvest the peritoneal fluid, what you see is a massive amount of neutrophils that infiltrated the organ. And in the right panel, you can see that it's PBS, and right is PBS and left is PS. And you can see that it's a massive amount of neutrophils detected by flow cytometry versus PBS. And then we calculate that it's about 10 million neutrophils in the peritoneum after the injection of the number of liposomes that we tell you. The infiltration is very robust, it's very rapid. We can see neutrophils in the peritoneum after 3 hours of the injection. You peak about 16, and they disappear after 3 days. So, if there was neutrophils in the peritoneum, and we know that these cells have high capacity to phagocytize pathogens, we hypothesize that the presence of these neutrophils induced by PS in the peritoneal cavity of mice should be capable to deal with a bacterial infection. And to make it very relevant, we decided to use a model of sepsis. And in model of sepsis, there is tremendous amount of bacteria, and there is no single bacteria, it's a multibacterial process. And this is this model called C-coagulation and puncture. The model is very simple. You have the animal, mice have a sickle that is equivalent to an appendix. We ligate it, so we create a tissue of necrosis, and we perforate it, and we squeeze the bacteria and the feces in the peritoneum. So now you have a pathogen that is multi kind of organisms, and also feces that are irritant. And that produces a very robust inflammatory response. And if you do the model right, you can produce mortality in these animals. And we have tailored this model to have mortality within about 80% mortality within 72 hours. So now if you pre-treat these animals with PS liposomes and allow them, the neutrophils to infiltrate, and you do the C-coagulation and puncture model, what we observe is a remarkable increase in survival, both in males and female mice, if they were pre-treated with PS liposomes, but no if you pre-treat them with PC liposome. So this is a quite remarkable recovery in this model. So why? Well, actually, we calculated the translocation of bacteria, the amount of bacteria in the peritoneum, or the amount of bacteria translocated in blood, and you can see that with PS, the amount is really very small, whereas with PC, you have the normal level that an animal that was just injected with PBS or produced after sham operation. And that suggests that, indeed, these neutrophils are clearing the pathogens. Well, also, this model produces a very strong inflammatory response. And here we show that if you treat these animals with liposomes, PS liposomes, the level of cytokines in all these cases is relatively small, very reduced, that animals will treat them just with PBS. So the idea is that these liposomes go into peritoneal macrophages, they activate these cells, they produce chemokines, chemokines attract neutrophils, and the neutrophils can resolve the septic insult. Now, the most remarkable observation that we had was this. So we inject mice with the two stereoisomers of phosphatidylserine, L and D. L is the active, the biological active, T is a non-biological active. And what we found is that we injected with them, their effect was exactly the same. Both liposomes protect mice, the inflammatory response that the cytokines or the markers that they use were exactly the same. Now, if these two lipids that are stereoisomers produce the same effect, that strongly suggests that the effect of PS liposome is not just due to the presence of PS, it's due to the negative charge of the phospholipid. So we believe there is two things that are very interesting for this. First, because the two stereospecific lipids produce the same effect, it's very unlikely that their uptake is by a receptor-mediated process because receptors are specifically stereospecific, and also it's very likely that it's produced by endocytosis, and because both of them have the negative charge, we believe that the negative charge is the actually driving force in the activation of the inflammatory response in the macrophages. So it's not just PS liposomes, it's negative charge liposomes, and we tried with different negative charge liposomes and the effects are very similar, although not totally identical. So in summary, what I want you to take a message is that the activation of cells by extracellular vesicles is a very complex phenomenon in which all the components may participate changing the biological process. So when you show you that when you take macrophages and you stimulate them with exosomes or extracellular vesicles from one cell, and you have an effect, this effect is the product of the lipids, the proteins, and the nucleic acids present in these vesicles. So it's quite complex and cannot be just analyzed in detail by just thinking that, oh yeah, all of them are doing the same thing. The other thing that is important is that phospholipids have a great impact on macrophages gene expression, and the charge of the phospholipid may be a critical factor in cell activation. Obviously, there are many questions to be answered. How negative charge phospholipids modulate gene expression? Well, there is an old idea that is that was called the intracellular negative charge effect of cells, and the idea is that if you increase the negative charge of membranes, these will attract positive charged proteins to the membranes and activate them. So what we think is happening is that these liposomes, when they are endocytized and they are target to endosomes, they fuse with the membrane of these endosomes and now increase the negative charge of the membrane, and these activate some cytosolic molecules that are attracted to them and activate the process. However, we cannot discard that the process is also mediated, at least in part, by the fatty acids, and we are doing experiments changing the fatty acid of these liposomes to analyze what genes are regulated by the fatty acids and what are regulated by the head of the phospholipid. So with that, I'm going to finish. Obviously, a lot of people have been involved in all these projects throughout the many years, but mainly this was done by my colleague in the lab, David Cowley. And here is my group. This is my favorite restaurant in San Diego, Berta's, who has a dish of every country in Latin America, and we always go there for celebrations of the lab. And with that, I would like to thank you for all your attention. Oh. And before that, I forgot this. This is very important. I'm organizing a meeting. I'm part of the Cell Stress Society International, and we have a biannual meeting, and the next meeting will be next year, November 2019, in the beautiful city of San Diego. We have a spectacular avenue, and this is a very exciting meeting that deals with the stress in many aspects, from cells to the environment. And I invite all of you to come to this meeting next year, next November. And with that, thank you, sir, very much for your attention.